And thank you, Vaksore sir, for kind introduction. First of all, I would like to thank the organizing team for inviting me to deliver the lecture on the bioinformatics. So I will directly start with my subject. The today's topic of the lecture is bioinformatics concept and applications. So in life sciences, this quantitation and quantitative tools we are using on the daily basis. So whatever practicals we are doing or research we are using some tools for the quantification purpose. For example, if you want to measure the pH of a solution, you use pH meter, or for the determination of absorbance, you use spectrophotometer. So in life sciences, these quantitation tools are highly important today. But apart from these quantitation tools, we also use quantitative tools. Now, what are quantitative tools? Quantitative tools are actually the softwares which we are using for the data analysis, the data that we gather from the quantitation tools, right? So both of these things are highly indispensable in modern biology. So bioinformatics is basically a discipline of quantitative analysis of information relating to biological macromolecules with the help of computers. Because when a large amount of data is present, it is highly impossible for us to analyze that data manually. And today we use computers for such a huge amount of data analysis. So, in computer means we use hardware as well as software part of the computer for the data analysis. So, in bioinformatics, we use computers for the analysis of the data of biological macromolecules. Okay, a detailed definition I will go through in next few slides again. Now, basically, the development of bioinformatics as a field, it is a result of advancement in both molecular biology as well as computer science over the past 30 to 40 years. We know that in almost 19th century, Frederick Mischer identified there is some material present inside the nucleus and he named it nuclein. And later on, people rename it according to its acidic nature, nucleic acid. Then by the Griffith experiment, then Avery, MacLeod, McCarthy experiment, and Hershey and Chase experiment, we concluded that DNA is a genetic material. So since then, our interest in the DNA is continuously rising, and that gives rise to a new branch of biology, molecular biology. So Watson and Crick they find out they model the DNA structure as double helix, and whatever development happens in the molecular biology in last 30 to 40 years has lead to the development of bioinformatics. So very first database in bioinformatics, it was constructed by Margaret Dayhoff in 1965. The name of the database was Atlas of Protein Sequences and Structures. So this is the first database ever constructed in the field of bioinformatics. Then in early 1970s, Brookhaven National Laboratory, they established a protein data bank. Now this protein data bank is a database of protein three-dimensional structures. So when this PDB was started, almost less than uh, few dozen structure were there. But today the size is almost around zero point 0.2 million. This figure here is updated now. Almost 2 lakh structures are there in the PDB. Then these are some historical milestone in the field of bioinformatics. Like first algorithm was constructed by Needleman and Wunsch in 1970. That was for global alignment. Then Chow and Fasman, they again created algorithm for secondary structure prediction of the proteins. Then in 1980s, GenBank database was established. And very famous tools that we are still using today, FASTA and BLAST, they were developed in that time by William Pearson and Stephen Erskine. We will go into that also. And then Human Genome Project was started. Actually, Human Genome Project provided a major boost for the development of bioinformatics because a huge amount of data was actually gathered through the project. And at that time, we consider the need of computer for the storage of this large amount of data. We require softwares for the analysis of those genomic DNA sequences. So here in this picture, we can see that how the developments in molecular biology goes hand in hand with the development in the field of bioinformatics. Like as I mentioned, in the year 1953, Watson and Crick they proposed the DNA double helix model and they received the Nobel Prize for that. Later on in 1955, Frederick Sanger he determined the protein sequence. First protein sequence that was determined it was insulin, and for this discovery, Sanger received Nobel Prize in the field of chemistry. Then in 1977-78, there were two DNA sequencing techniques were developed. One was developed by Maxam and Gilbert, and another one was developed by Sanger, that we call as Sanger dideoxy DNA sequencing method. So again, for this discovery, Sanger received Nobel Prize in the field of chemistry. So Sanger is a person. He's a he's the second person who received Nobel Prize in the same field. Another person is from physics. Then in 1985, PCR was invented by Carey Mullis. So these are the developments in the field of molecular biology. Now see how developments in the field of bioinformatics also took place along with the molecular biology. So as people started to determine the protein sequences, there was a need of a protein sequence database. And Margaret Dayhoff, she established that database. The name was Atlas of Protein Sequences and Structure. So this database comprises of protein sequences as well as protein structure. Then in 1970, the sequence alignment algorithm was developed. So the sequence alignment is a process in which we try to find out the matches and mismatches in between the two or more sequences. Then early internet was started in year 1969. It was called as ARPANET, ARPANET. So that early internet was actually developed for the communication in between the different bases of the US military. And later on, that was developed into the internet that we see today. Then PDB was established by Brookhaven National Laboratory, that is Protein Data Bank, which comprises protein structures. Because up to that time, we were able to determine the protein structure by X-ray crystallography method. And today we are determining them by NMR, that is nuclear magnetic resonance, as well as cryo-electron microscopy, etc. Then in 1980s, a database was established. The name is GenBank. So it is a nucleotide sequence database. So as Sanger dideoxy method and Maxam and Gilbert method were developed, people started to determine the DNA sequence. And in order to store those DNA sequence, there was a requirement of nucleotide sequence database. And that's why GenBank database was established in USA. Then in late 1980s, Human Genome Project was initiated. Then first bacterial genome sequence was determined in 1995. So the bacteria was Haemophilus influenzae. The first genomic DNA sequence that was determined it was 5X174. The first bacteria whose entire genome sequence was determined it was Haemophilus influenzae. Later on, yeast genomic DNA sequence was determined. And in 2001, first human genome draft was published. And in 2003, complete human genome was published. So these are the advancement in the field of molecular biology. Similarly, you can see in the late 1980s, SwissProt database was established. So SwissProt database is specifically dedicated for the storage of protein sequences. Then there was establishment of NCBI in USA, that is National Center for Biotechnology Information. Now this institute create new databases, new software tools for the bioinformatician or for the uh, people in the field of life sciences. Then FAST-A tool was developed. So FAST-A tool was developed by William Pearson. It is a database similarity search tool. Then BLAST tool was developed at NCBI by Stephen School in 1990. It is also a database similarity search tool. Then Internet was started in 1991, that is World Wide Web. And in 1992, EBI was established, that is European Bioinformatics Institute. So in these two slides, I hope you understand that how the development in the field of molecular biology and bioinformatics goes hand in hand. As molecular biology developed, there was a growing demand of the storage system, and that was provided by database. Again, for the analysis of that data, there was a requirement of software. So those software again provided by bioinformatics. I hope I'm clear up till now. So, after Sanger's discovery of nucleotide sequencing or DNA sequencing, people tried to automate that technique. And the company Applied Biosciences, they develop a automated DNA sequencer based on Sanger sequencing method. And the machine itself is called a Sanger sequencer. So this is the image of Sanger sequencer. You can see there are multiple Sanger sequencers in a human genome determination laboratory or human genome project laboratory. Almost 200 machines, they work for 24 by 7 in order to determine the human genome. So this is the era of 1990s where a lot of genomic DNA sequences were determined. And at that time, we were using this Sanger sequencer machines. Nowadays, we are using next generation sequencing platform like Illumina, Ion Torrent, like that. So because of automated DNA sequencer, the amount of or the number of sequences and the number of base pairs of the DNA, they increase exponentially. So in this period of 1980s to 1995, you can see the line is almost flat on the graph, because in this period majority of the DNA sequencing was carried out by the old traditional method of the Sanger DNA sequencing, where you have to use the radio labeling, then you have to do the page, that is polyacrylamide gel electrophoresis, then you have to do the autoradiography. So in that particular experiment, you cannot determine more than 500, 600 base pair sequence, and it took a lot of time. But as soon as the automated DNA sequencers came, many people started to determine the gene sequence or the genomic DNA sequence, and that's why the amount or the number of sequences, they have increased exponentially. So number of sequences, that figure is in million, and the number of base pair, that figure is in billion. So today we talk about genomics, proteomics, metabolomics, or transcriptomics. So in genomics, we try to address the entire genome. We try to determine the entire genome sequence, as well as we determine the position of the gene and the function of the gene, right? So in order to determine the genomic DNA sequences, we have this next generation sequencing platform. So in this slide, you can see the size of the genomic DNA sequences of the different organisms. Like a simple bacteriophage, T2 bacteriophage, it has a genome size of 1,70,000 base pair. Then a simple bacteria E. coli, its genome size is around 4.6 millions of base pair, means 46 lakh base pair. Then Drosophila melanogaster, which is a model organism for the insect, 130 million base pair. If you talk about a human, the genome DNA size is 3.2 billion base pairs. Means a single cell, a haploid set of chromosome consists of 320 crore of base pairs. And that sequence we have already determined. So for many of the prokaryotes and many of the eukaryotes, we have already determined the genomic DNA sequence. Then canopy plant, Paris japonica, 150 billion base pair. So in this graph, you can see as the complexity of the organism is increasing, the genomic DNA size is also increasing. Like for the viruses, the genomic DNA size is ranging in between 5,000 base pair to almost around 1 lakh base pair. For bacteria, it is from around 10 lakh base pair to 100 lakh, means 1 crore base pair. For fungi, it is even more higher. So on the y-axis, you can see KB, MB, and GB. So KB stands for the thousands. MB stands for the million of base pair. It is called as mega base pair. Mega base pair means millions of base pair. and GB stands for giga base pair. So giga base pair denotes the billions of base pair. Okay. So you can see almost all the higher organisms like mammals, reptiles, plants, amphibians, they have this size in GB, starting from almost around uh, 1 GB to 100 GB, means 1 billion base pair to 100 billion base pair. So we have already determined the genomic DNA sequence of many organisms. So a few hundred of eukaryotes, thousands of prokaryote genomic DNA sequence have already been determined by us. So what we do in bioinformatics basically, in biology, whatever sequence and structure data we have, it is highly complex. Because if you just see the example of a human whose genome size is 3.2 billion base pair, 320 crore base pair, that's a huge amount of data to analyze manually, right? Also, the structural data that we have, also the structural, yeah, also the structural data that we have of the protein structure, it is also a very complex kind of data. So it is impossible for us to analyze this data manually. So what we do, we try to analyze this complex data with the help of computer softwares. And we transform that complex data into knowledge. And that is bioinformatics. So why bioinformatics has emerged as a very important branch in the field of biology? Because enormous amount of biological data has been synthesized in terms of sequence data as well as structural data. So there was a demand for efficient computational tools to manage and analyze the data. We wanted a highly efficient computers, highly efficient softwares which can manage that data, which can store that data and which can analyze the data, and it can transform. We can transform that data into knowledge. Then the development of this computational tool depend on knowledge generated from the wide range of disciplines, including mathematics, statistics, computer science, information technology, and molecular biology. So basically, bioinformatics is a multidisciplinary field which incorporate mathematics, statistics, computer science, information technology, as well as molecular biology. Now, the merger of these all disciplines created an information-oriented field in the biology, which is known as bioinformatics. So as I mentioned, that bioinformatics touches all the branches like math, stat, engineering, chemistry, biochemistry, computer science, etc. So in this slide, you will learn the definition of bioinformatics. Bioinformatics is an interdisciplinary research area at the interface between computer science and biological science. This is the proper definition of bioinformatics. Bioinformatics can be defined as a union of biology and informatics. Bioinformatics involves the technology that uses computer for storage, retrieval, manipulation, and distribution of information related to biological macromolecules such as DNA, RNA, and proteins. So, if I elaborate this definition, today, whatever data we have, whatever kind of data, not only biological data, but whatever data we face in our day-to-day life, we capture that data or we store that data into our mobile phones. So mobile phones are also mini computers. They also have RAM, ROM, CPU, input and output devices. So each type of data we are storing in the computer, right? So what the data is stored in the computer today. Second thing is, once we store the data in the computer, we also retrieve that data. Retrieval means to get back. Because when we save a file, तुम्ही जेव्हा फाईल सेव्ह करतात कम्प्युटरमध्ये एखादी, तुमचा पर्पज फक्त सेव्ह करणे नसतो. त्या फाईलला पुन्हा कधीतरी तुम्हाला ओपन करायचं असतं, तिला एडिट करायचं असतं, तिला अनालाइज करायचं असतं. सो त्या रिट्रायव्हल साठी सुद्धा आपण काय वापरतो? यूज कम्प्युटर फॉर रिट्रायव्हल पर्पज, राईट? वी गो टू दॅट फोल्डर, we search for the file, and then we double click on the file, and then that file opens up. This process is called as retrieval, right? Third thing is manipulation. So manipulation is like analysis. Suppose I have to draw the standard curve. समजा मला एक मी ऍसे करतोय बायोकेमिकल ऍसे मे बी शुगर एस्टिमेशन आणि मला एक स्टँडर्ड कर्व काढायचा आहे. So majority of the people, what they are doing today, they are putting those value into Excel, and in the Excel they are drawing that scatter curve, and then they draw the trend line, and then you get the equation. R square व्हॅल्यू तुम्हाला मिळते, रिग्रेशन लाईन तुम्हाला मिळते, आणि त्याद्वारे मग तुम्ही अननोनचे कॉन्सन्ट्रेशन डिटरमाइन करू शकता. सो दिस इज व्हॉट मॅनिप्युलेशन इज. दिस इज अनालिसिस. So today, if you want to analyze large amount of data, we are using computer for that purpose. And fourth thing is distribution. Because whatever data we are having today, we do not want to keep it only to ourselves, but to distribute into the scientific community. And today, for the distribution or for the communication of anything, you're using internet, right? You're using social media platforms, or you're using some kind of websites for the communication of the data that you have, right? And what is internet? Internet is a network of computers, right? So for everything today, we are using computers. Similarly, in the field of biology or in the field of molecular biology, when you have data of DNA, RNA, and proteins, then and we know that the data of this molecule is very large. जसं मी तुम्हाला उदाहरण सांगितलं की बऱ्याच अशा स्पेसिस आहेत बॅक्टेरियांच्या ज्यांचा जिनॉमिक डीएनए सिक्वेन्स आपण डिटरमाइन केलेला आहे, आणि त्यांची साईज ही काही लाख बेस पेअर मध्ये आहे. जसं इकोलायची जिनॉमिक डीएनए साईज 46 लाख बेस पेअर, ह्युमनची जिनॉमिक डीएनए साईज 320 कोटी बेस पेअर आहे. म्हणजे मुळात आपल्याकडे खूप मोठ्या प्रमाणात, प्रचंड प्रमाणात हा डेटा, सिक्वेन्स डेटा ऑलरेडी उपलब्ध आहे, आणि त्याला स्टोअर करण्यासाठी कम्प्युटरपेक्षा दुसरं कुठलंही चांगलं माध्यम आज उपलब्ध नाहीये. आणि म्हणून 4 शब्द मी पुन्हा वाचतो. स्टोरेज, सिक्वेन्स डेटा असेल किंवा स्ट्रक्चरल डेटा असेल, आपण कम्प्युटरचा वापर करून त्याला काय करू शकतो, स्टोअर करू शकतो. सेकंड थिंग इज रिट्रायवल, वी कॅन अगेन गेट बॅक दॅट डेटा. थर्ड थिंग इज मॅनिप्युलेशन और अनालिसिस, जो काही डेटा आपल्याकडे आहे त्याचं अनालिसिस आपण करू शकतो विथ द हेल्प ऑफ कम्प्युटर. अँड डिस्ट्रीब्युशन, म्हणजे मला कम्युनिकेट करायचा हा डेटा, तर मी ऑनलाईन डेटाबेस मध्ये तो टाकून देईल, आणि जगामध्ये कोणीही तो डेटाबेस जर ऍक्सेस करू शकत असेल तर त्याला तो डेटा मिळेल, राईट? So this is the definition of bioinformatics. It can be defined as union of biology and informatics. Bioinformatics involve technology that uses computers for storage, retrieval, manipulation, and distribution of information related to biological macromolecules such as DNA, RNA, and proteins. Now the question arises, if I say biological macromolecules, then why only these three macromolecules are considered, DNA, RNA, and protein? Why not the carbohydrates are included, or why not any other molecule is included? The reason is DNA, RNA, and proteins, these molecules are information rich. None of the other molecule have information. The sequence of DNA is not random, or the sequence of RNA and protein is not random. It has some meaning to the cell, right? So DNA, the triplet codons in the RNA, it code for a particular amino acid. Now that code language is understood by the cell, and it is universal. The same codons are there in all living organisms, right? Now with because of Hargomin Khurana, Marshall Nirenberg, and Robert Hole, we also now know the genetic code, that there are 64 genetic codes, and which code is coding for which amino acid, right? So only these three molecules has information, and that's why in bioinformatics we only focus on these three molecules. Then bioinformatics has two subfields. These are the two subfields of bioinformatics. This is the foundation. You can see at the base, this is the foundation of bioinformatics. It includes software development and database curation and construction. So, if you suppose you determine a particular gene sequence. Now, if you want to save that gene sequence into database, there should be a database in first place, right? So somebody, you cannot create database because you're a biologist. So there should be somebody who construct that database where a biologist can store the sequence, or from where the biologist can retrieve a sequence, right? So this is the foundation of bioinformatics. where people from computer science or software engineers or the people from life sciences who have learned the programming languages, they develop softwares, they develop databases. Without software, without databases, we cannot uh, save the data into database, or we cannot analyze the data, right? So that's why this subfield is highly important. Software development, database construction, and curation. Then there is a second subfield. Now, second subfield is called as application. Now, there are three different types of applications in bioinformatics. One is sequence analysis, function analysis, and structural analysis. Now, these analysis can be done by the software. We can do the we can analyze sequence, we can analyze function and structure with the help of a software. Also, the data that we have, we can store into a database, or the data that we want, we can retrieve from the database. So that's why I'm saying again and again that this foundation is very important. Somebody should have created a software, somebody should have created a database so that we can use it. So majority of the biologists come into another subfield, or they are the part of another subfield, that is application part. आपण अनालिसिसच्या पार्टमध्ये मोडतो, कारण आपल्याला प्रोग्रामिंग लँग्वेजेसचं नॉलेज नसल्यामुळे वी डू नॉट डेव्हलप सॉफ्टवेअर, और वी ऑल्सो डू नॉट डेव्हलप डेटाबेसेस. जे आधीपासून बनलेले सॉफ्टवेअर आहेत, जे बनलेले डेटाबेसेस आहेत, त्याचा यूज आपण करतोय. मग आपण कशासाठी यूज करतोय, तर सिक्वेन्स अनालिसिस. याच्यामध्ये महत्वाचं अनालिसिस आहे सिक्वेन्स अलाइनमेंट, देन डेटाबेस सर्चिंग, मोटिफ डिस्कवरी, जीन अँड प्रमोटर प्रेडिक्शन, phylogenetic analysis, where you can study the evolutionary relationship, genome comparison. Then function analysis include protein subcellular localization prediction, protein interaction prediction, gene expression profiling, metabolic pathway modeling. And in structure analysis, we do the protein structure comparison, protein structure classification, protein structure prediction, and nucleic acid structure prediction. So these are the different type of analysis we do by using softwares and databases that are already developed. Now, the bioinformatics has wide set of applications. It has applications in pharmacogenetics, pharmacogenomics, in immunoinformatics, proteomics, chem informatics, medical informatics, computational biology. Everywhere it has its own applications. So, why do we use bioinformatics? The question is, as a person from a life science, how bioinformatics can benefit me, or why should I use bioinformatics? The first important reason is you can store biological information there. The databases that are developed in the field of bioinformatics, we can store our own data into those databases, or we can retrieve the data from those databases. Then we can retrieve and compare the gene sequences. So, suppose if there is a person who is working at the molecular level and they determine a gene sequence or a protein sequence. Now they want to compare their own sequence with the sequences from the database. That you can do. We can predict the function of an unknown gene or unknown protein. So suppose you determine a gene sequence or a protein sequence, but you do not know its function. So with the help of bioinformatics tools, we can predict the function of that molecule. Then we can search for previously known functions of the gene. We can compare data with other researchers, as well as we can compile and distribute data for other researchers. Then comes the application part. A very important application today is in the field of medicine, knowledge based drug designing. Now, earlier, when the pharmaceutical companies tried to develop a new drug, it took around 10 to 12 years and billions of dollars of expenditure there. But today, with the help of bioinformatics and molecular docking tools, there is lot of less time is required, and in the less money you can develop that drug. That is called as a knowledge based drug discovery. You just have to have a protein structure, the target protein that you want to inhibit or that you want to activate. You just have to have the structure of that protein, and you just need a chemical compound library, that also virtual. And with the help of docking tools, we can check whether the molecules from my library are attaching with the target protein or not. So that's a very important application there, where the time has been reduced for the new drug discovery as well as cost have been reduced. Then forensic DNA analysis. There is very famous example of CODIS database. Now this CODIS database was developed by FBI, that is Federal Bureau of Investigation of USA. And what they have done, they have stored the DNA fingerprints of all the criminals that were caught. जसं बोटांचे ठसे घेऊन ठेवले जातात यांचे गुन्हेगारांचे किंवा फोटोग्राफ्स घेऊन ठेवले जातात, तसं FBI ने काय केलंय, त्यांच्याकडे जे कोणी क्रिमिनल अरेस्ट होतील, त्यांच्याकडे जे कोणी क्रिमिनल असतील तसं DNA फिंगर प्रिंटिंग करून त्यांनी ठेवलंय. आणि ते सर्व DNA फिंगरप्रिंट्स त्यांनी एका डेटाबेसमध्ये स्टोअर केले कोडिस नावाच्या. सो आता जेव्हा केव्हा फ्रॉम द क्राईम सीन दे गेट द ब्लड सॅम्पल ऑर अ DNA सॅम्पल, आणि ते DNA सॅम्पल त्याचा DNA फिंगरप्रिंट जर व्हिक्टिमशी पण मॅच होत नसेल किंवा सस्पेक्ट बरोबर पण मॅच होत नसेल, तर तो DNA फिंगरप्रिंट ते चेक करतात की कोडिस डेटाबेसमध्ये कोणाशी मॅच होतोय का. आणि जर तो मॅच झाला कोडिस डेटाबेस मधल्या कुठल्या क्रिमिनल बरोबर, म्हणजे तो क्रिमिनल त्या स्पॉटवरती प्रेझेंट होता हे सिद्ध होतं. So that's a very unique application of this bioinformatics in the forensic DNA analysis. Then agricultural technology. Now we have genomic DNA sequences of majority of the commercially important crop plants or agronomically important crop plants. So with the help of genomic DNA sequence, we can easily identify the genes which are responsible for a particular trait, right? And that is how we can develop new varieties, or we can develop the transgenic crop plants also. So that has a tremendous application in agricultural biotechnology also. Then in molecular phylogenetics. So nowadays we are determining the DNA sequence, and with the help of those DNA sequence we try to identify a particular species. So that is a molecular phylogenetics. So even in case of SARS-CoV-2, that is COVID virus, when it was initially identified in the China, they also identify it with the help of molecular phylogenetic technique. They determine the RNA sequence, then they compare it with the database, and then they created a phylogenetic tree, and they concluded that this virus belongs to SARS family, but it's a unique virus. Its entire sequence is not matching with existing virus, so they name it SARS-CoV-2. Then the structure here you can see on the slide. This is the spike protein structure of coronavirus. Now, this protein is actually responsible for adherence of the virus to the lung cells, and that is how it enter into the lung cells. So this protein is actually responsible for the infection. Now, immediately when the corona was started in December 2019, this structure was determined in January 2020. Scientists determined this protein structure in January 2020, and immediately by using this structure, there is a application of bioinformatics in the vaccine development also, vaccinology. So with the help of bioinformatic tools, we can predict where the B cell epitopes are present and where the T cell epitopes are present. That is called as epitope prediction. So by using this structure, they predicted the epitope, and that gave them insight for the development of vaccine in a very short amount of time. Okay, so in vaccine development also, this bioinformatics has a very huge application in terms of epitope prediction. All right, so that was just a introduction to bioinformatics. We will now see the databases. What are databases and various types of databases that are there in bioinformatics. So what is a database? Database is actually a structured collection of the data held in computer storage, especially one that incorporates software to make it accessible in a variety of ways, or any large collection of information is called as database. So database basically is a computerized archive. It is a computerized storage system where the data is not stored randomly, but the data is stored in some structured manner. some ordered fashion. Because whenever next time somebody want to retrieve a particular data, it should be easy for them to retrieve it. So that process is called as making a query, right? So for example, nowadays colleges are also maintaining their own database of the students. They fill up online application form or online admission form, and entire data of that student is stored into the college database. So whenever next time if you want to pull out information of a particular student, you just give them SID number, and for that SID number, whatever information is available in the database, that will be retrieved. Or if you don't have a SID number, you can simply retrieve the information with the help of phone number, email ID, address. So whatever query you give to the database, for that query, whatever information is present in the database, that will be given to the user. That is the beauty of database. So database management system is the organization and manipulation of data in the database. So there are software packages which actually use as a database management system. For example, there is SQL is there. Then there is C++ we can use, which is a programming language for development of object-oriented database management system. Or SQL, that is structured query language, is used for the development of relational databases. So as I mentioned, that the data in the database is highly structured. Structured means what? Suppose you go to the library, the books are not randomly arranged. There is also they are maintaining some structure. Like there is a separate cupboard for a specific subject. Every book is given with some accession number. So as like library, here also in the database we store these sequences or files in some structured fashion. Then databases are searchable. Some index is given to the database so that you don't have to search through entire database, but you can find out a particular piece of information just by providing a query. Then these databases are updated periodically, and databases are cross-reference. Their cross-reference means databases are interlinked together. Like DNA sequence data will be interlinked with the protein sequence data, or protein sequence data will be interlinked with the protein structure data database, or all these databases are interlinked with some literature database like PubMed, like that. It include associated tools like software which are necessary for access, updating information, insertion, and information deletion. And data storage management system, as I mentioned, there are three different types of database management system: flat file system, relational database, and object oriented database. Now, why there is a need of biological databases? आता बायोलॉजीमध्ये डेटाबेसची काय आवश्यकता भासली, तर याचं कारण आपण आता आधीच्या काही स्लाइडमध्ये पाहिलं, की खूप मोठ्या प्रमाणात डीएनए आरएनएचे सिक्वेन्स डिटरमाइन करण्यात आले. खूप मोठ्या प्रमाणात प्रोटीनचे सिक्वेन्स आणि प्रोटीनचे स्ट्रक्चर डिटरमाइन करण्यात आले, अँड दॅट लीड टू द एक्सपोनेन्शियल ग्रोथ इन द बायोलॉजिकल डेटा. आणि मग एवढा मोठा डेटा साठवायचा कुठे, ठेवायचा कुठे, त्याला स्टोअर कुठे करायचं? मग त्यासाठी आपण डेटाबेसेस बनवले, कम्प्युटराइज डेटाबेसेस बनवले, जे खूप मोठ्या प्रमाणात डेटा स्टोअर करू शकतात. आता बायोलॉजीमध्ये सिक्वेन्स किंवा स्ट्रक्चरल डेटा सोडून आणखी कुठल्या प्रकारचा डेटा प्रेझेंट असतो? तर सिक्वेन्स एक आहे, 3D स्ट्रक्चर ऑफ प्रोटीन एक आहे, 2 डायमेन्शनल जेलच्या इमेजेस, दॅट इज ऑल्सो वन टाइप ऑफ डेटा. देन MS अनालिसिस, दॅट इज मास स्पेक्ट्रोमेट्री अनालिसिस जे आपण करतो, मॉलिक्युल्सचं म्हणा, प्रोटीन्सचं म्हणा, त्याचाही डेटा एक आहे. Then you do study of a gene expression by DNA microarray, त्याचाही डेटा जनरेट होतो. So all these data today, they are not published in a conventional manner, but they are directly submitted to the database. म्हणजे तुम्ही एखादं रिसर्च आर्टिकल जेव्हा पब्लिश करणार, त्याच्यामध्ये जर तुम्हाला जीन सिक्वेन्स पब्लिश करायचा आहे, तर तुम्ही तो जीन सिक्वेन्स आर्टिकलमध्ये नाही लिहित. तुम्ही तो जीन सिक्वेन्स डेटाबेसमध्ये सबमिट करतात आणि त्याचा ऍक्सेशन नंबर फक्त तुम्ही आर्टिकलमध्ये लिहितात. सो एखाद्याला तो डिटेल जीन सिक्वेन्स बघायचा, तो ऍक्सेशन नंबरने त्या सिक्वेन्सला ऍक्सेस करू शकतो डेटाबेस मधून. अँड नाउ इट इज मॅन्डेटरी. जर तुम्हाला एखादा जीन सिक्वेन्स, प्रोटीन स्ट्रक्चर पब्लिश करायचं, त्याच्या संदर्भातली माहिती पब्लिश करायची आहे, तर आज जर्नल्स तुमचा पेपर ऍक्सेप्ट करणार नाही जोपर्यंत तुम्ही तो सिक्वेन्स किंवा स्ट्रक्चर डेटाबेसला सबमिट करून त्यांच्याकडून ऍक्सेशन नंबर घेत नाही. So it is now mandatory to submit this sequence and structures to the database and to get accession number from them. So now this is how the biological data was distributed over the period of time. Like initially it was distributed in the field of in the form of books and articles, then in the form of computer tapes, then floppy disk, CD-ROM, FTP is still use we are using today, that is file transfer protocol. Online services, then www, that is world wide web, means internet. We are using internet still for the distribution of data and DVDs. These are some statistics about the databases. There are more than 1000 different biological databases are present in the world. Their sizes are variable. Some are less than 10, 100 KB, some are more than 20 GB. For example, DNA sequence data is more than 20 GB, protein sequence data is around 1 GB, 3D structure data is there, and other very smaller databases are present. Some databases they are updated on the daily basis. Some databases are updated on annual basis. Some databases does not update at all. And usually these databases are accessible through web. Some are free and some are paid databases. So these are the name of some of the important databases in the world. Now, as I mentioned, there are different categories of the databases in life sciences. For example, there are sequence databases. It include nucleotide sequence database and protein sequence databases. Then there are genomic sequence databases. There is mutation or polymorphism database. A very famous example is OMIM, Online Mendelian Inheritance in Man. That is a mutation or polymorphism database where the different allelic information and their sequence information is stored. Then protein domain and family databases are there. These are generally secondary databases. For example, Prodome, PFAM, Print. These are all examples of the protein domain and family databases. Then proteomic databases are there, which incorporate the 2D gel image data, mass spectrometry data. Then 3D structure database is there. A very famous example is a PDB, Protein Data Bank. Then metabolic network database is there. The name is KEGG, KEGG, KEGG database. It includes all the metabolic pathways of the living organisms that are known today. Then there is regulatory network database, bibliographical database, where the books, research articles, review articles, their citation is stored. Example is PubMed. Then expression database, where the gene expression data is stored. For example, microarray data. And there are some specialized databases are also present, which serve a particular scientific community or a specific model organism. So this is the homepage of NCBI. So if time permit, I will also show you some practical of retrieval of the data from the databases, as well as sequence alignment and database similarity searching. So this is the homepage of NCBI, that is National Center for Biotechnology Information. Now this institute of USA was established by NIH, National Institute of Health. They have established NCBI. Now what is the function of NCBI? So NCBI hosts different databases, biological databases, and they also develop new databases according to the need of people in the field of molecular biology. They also host some popular resources or popular tools or softwares that are routinely used by bioinformaticians. For example, BLAST. Now, BLAST is a computational tool used for uh, database similarity searching. Now, its link is available on the NCBI. There are many databases. If you click on this all database list here, you can read the databases that are hosted by NCBI. It includes GenBank, it includes a protein sequence database, it includes PubMed, it includes OMIM database, it includes genomic DNA sequence database. So a lot of databases are hosted by NCBI. And there are multiple tools also, like BLAST. Then PubMed. PubMed is again a bibliographical database or a citation database, which is a part of Medline. So Medline covers the field of medicine, nursing, dentistry, veterinary medicine, public health, and preclinical sciences. They contain citations from approximately 5,200 worldwide journals in 37 languages. And they have almost around over 20 million citations since 1948. This much large amount of bibliographical data is present in the PubMed. So this is how we can search the PubMed for the topic of our interest. So suppose somebody want to do research on mitochondrial evolution and he want to find out what are the research articles published on the mitochondrial evolution. So you just put the keyword here in this search box and click on search button. You will see the list of articles where the mitochondrial evolution keywords are present. Now, if you want to read the abstract of this article, you can simply click on this, the name of the article or the title of the article. This is how you can watch or you can read the abstract. Now, if the article is useful to you, then you can download the full article from here. You can see on the right hand side, there is a full text link is present. You can click here and you can download the full text article if it is freely available. So just simply type in the query and click on the go button. You can access the article. Now, biological databases are actually divided into three categories: the primary database, secondary database, and a specialized database. We will see the difference in between them. Now, primary database contain original biological data. Now, what is the meaning of original biological data? Original biological data means raw data, where it doesn't have any kind of processing. So, suppose you determine the sequence of a DNA or a sequence of a protein, that is a raw data. You haven't done any kind of analysis there. So that is called as original biological data. They are archives of raw sequences or structural data submitted by the scientific community. So in primary databases, anybody can submit their data. They will accept. Now suppose I am working on an insulin gene. Few other people are also working on an insulin gene, and we determine insulin gene sequence. And all of us submit that insulin gene sequence to the GenBank, right? Now, GenBank will not say no to anybody. They will accept the insulin sequence from all the people and store it. So that leads to the repetition of the data. Now, if you search GenBank, you can observe a lot of insulin gene sequences there. That leads to redundancy of the database. Means primary databases are redundant. The repetition of data is there. Similarly, PDB is also a primary database for protein structures. Then comes secondary databases. Now, secondary databases contain computationally processed or manually curated information. So here in secondary databases, we as a part of scientific community cannot directly submit the data. The data present in the secondary database comes from primary databases, but it is only submitted by the database operator or database curators. So suppose in the secondary database, they want to submit the insulin gene sequence, then they will take that sequence from the primary database itself. If there is an error, they will remove the error. If there is any annotation is required, they will do the annotation and then only they can submit. They will submit a sequence into secondary database. So secondary databases will not be redundant as like primary database. Means in secondary databases, there will not be any kind of repetition of the data because nobody is allowed to submit. Only database curators can submit the data there. So that's why there is no repetition. Then comes specialized databases. Specialized databases are those that cater to a particular research interest. For example, Flybase. Now, Flybase is a database which is dedicated to Drosophila melanogaster. So, the people worldwide who are working on Drosophila, for them, this database is highly useful. Then HIV sequence database. So, the people or the scientists who are working in the field of AIDS or on HIV, for them, this database is very useful. Then ribosomal database project, they are databases that specialize in particular organism or particular type of data. They are called as specialized databases. So this primary databases, it include GenBank, DDBJ, that is DNA Data Bank of Japan, EMBL, that is European Molecular Biology Laboratory. Then in protein sequences, there is SwissProt, Tremble, that is translation of EMBL, PIR, that is Protein Information Resource, UniProt. These are some protein sequence databases. And in case of structure, there is only one primary structural database is present, that is PDB, Protein Data Bank. So in primary databases, there are three major public sequence databases are present. One is GenBank, another one is European Molecular Biology Laboratory, that is EMBL. Now the EMBL name has been changed recently. It is called as ENA, that is European Nucleotide Archive. Okay. Then there is a DNA Data Bank of Japan. These three databases are freely available. So a small number of sequences, especially those published in the 1980s, were entered manually from the published literature by the database management staff. But today, it is mandatory to submit a sequence into these databases. So these three databases, they collaborate and exchange new data on the daily basis. So these three databases, they have done a collaboration. The name of the collaboration is International Nucleotide Sequence Database Collaboration. According to this collaboration, every database has to share the information of the sequence data with other two databases on the daily basis. Okay.
Excuse me, sir.
So, yes, yes.
Your presentation not seen, sir.
Is it problem with everyone?
We are able to see. We can see. I can
Yeah,
say.
yeah. Because also I have another screen now. I can see that. Maybe some connection issue with people. You just leave the meeting and rejoin.